Okay. Buongiorno a tutti. So, good morning, and I'm pleased to welcome you here at the Edmund Mack Foundation for this PhD Day 2023. And I bring you the warmest welcome from the President of the Foundation, the Board of Directors, and the entire Foundation. And I would like to thank the colleagues that have contributed to the organization of this day. And thank you for giving me the opportunity to say a couple of words as introduction and to state once again the importance of the PhD program for the Foundation. And I would like to connect this importance of the PhD program to the history of this institution. In fact, we are approaching a very important anniversary for the Foundation. Soon it will be 150 years from its institution. And it was back in 1874 when here in San Michele all'Adige, an agricultural school with a so-called at that time experimental station was established here. And this has been an institution that has been spanning the course of history, first becoming the Istituto Agrario di San Michele all'Adige and nowadays the Edmund Mack Foundation. And all these 150 years of research and experimentation help us, on one hand, to keep a clear focus on innovation as a tool to face the challenges that the agricultural world and our territory are facing and to ensure its competitiveness. On the other hand, these 150 years of history have taught us very important values. And one of these is the importance of being humble but determined in pursuing our objectives. Because the more we learn, the more we deepen our knowledge, the more we realize how broad the knowledge of nature is and how much our knowledge is still just a small part of the greater knowledge. And knowledge has always been here the key word in facing and overcoming challenges. And in fact, the founding idea of this institution has been, since the beginning of that vocational school and the experimental station, to bring together education and research. And this idea is still one of the founding pillars of the Foundation. And we can see in our PhD program the transposition in modern times of that original idea that has become today to bring together the most advanced research with the highest education level. And the commitment of the Foundation to this program, I believe, finds concrete evidence in the numbers of the program itself that I think will be soon presented by Professor Pezzotti in a few minutes, and the numbers of the students, the number of the scholarship that we are funding, the wide national and international network of research centers, universities, and private companies that we involve in the program, the time and the energy that our researchers put in supporting the students. And all this is resulting in a high quality work that you all students are delivering. Now, I don't want to take additional time to this day, which is dedicated to the student community and is an important moment for you all researchers and students to share progresses, experiences, and ideas. So thanking the University of Trento for the collaboration in organizing this day and all the scientists and researchers that will contribute to the session and discussions today, I wish you a very productive day, and I thank you so much for the attention. Have a good day.
I don't know if I need to speak on the speaker. Maybe it's better to speak on the speaker. So I would like to invite, thank you very much, Mario, and I would like to invite Professor Riccardo Ricon to some greetings from the University of Trento. And Professor Ricon is now the director of the C3A, so representing the University of Trento. Please.
Good morning, everyone. As Professor Pezzotti told, I am the director of the Center of Agriculture, Environment and Food of the University of Trento. Maybe some of you doesn't know. Maybe some of you doesn't know what is the center, but the center was born together with San Michele Foundations and and Fondazione MAC, and is now going to a new age in which the collaboration, hopefully with the foundation, will grow in the 3 or 4 the 3 or 4 topics on which we are working, which is agriculture, looking to new foods, looking to the Great New Deal of the environment, and looking at the socio-economic part of the involving agriculture. I am also talking as a former coordinator of the doctoral program, which is in which involve almost 70 students in the 3 years, almost 4 years now, due to the COVID, and is a group of people of which are you, but of which we are very proud. And I also introduce you Gerardo Popolo, which is the new coordinator. And through when I see you, I see the students, I see the future not only of our center, but also of research and research also of Italy and the world. So I think this is one of the best moments of the year when I can talk to you, see you all together, and all together we can discuss of science.
Thank you very much, Riccardo, and this is very important for your nice words and for the vision that you gave to the relationship between the University of Trento and Fondazione MAC. I would like to say some few words about the PhD communities. So, an institution of research without PhD students is without any beating heart. So without it's mutilated. So it is very important that the PhD community would be relevant, would be present, would be active, would be the beating heart of the Fondazione as a research and innovation center. Just because without PhD student means that there are no ideas, there are no original ideas, there are no projectuality, and there is no way of growing or developing. So I would like very much to say that the PhD students are always welcome to come to visit me for whatever they need, for whatever they want, and I will always support them in their progress during their courses and their projects, their research projects. This is something that every PhD student should know, that in University of Trento and Professor Gerardo Popolo and myself, they will find help for discussion, help for envisaging, help for directing them together with the scientists inside and professor inside the Fondazione Centro Ricerca Research Innovation Center. I would like to say that this beating heart is about 80 students. They are enrolled partially at the University of Trento, but are enrolled in many universities in Italy and some universities abroad, in Europe and in the United States and in New Zealand. So we are spanning from all over the world. So this is very important. The internationality of the community is very important for enlarging ideas. So I would like very much now to introduce the speakers, the keynote speakers. As Riccardo said, there are three elements of that we say: agriculture, agrosystem, biodiversity, and environment. So the speakers that we have today are Professor Enrico Pe from Scuola Sant'Anna. He's a friend, but he's a geneticist that would talk about his experience in doing breedings for undeveloped countries. And then we will have the hydrologists. The hydrologists we have Professoressa Maria Cristina Rulli from Polytechnic of Milano, and she will be introduced later by Riccardo, and then she's working in assessment according to global change scenario, hydrology, river geomorphology, hydrology, and slope stability. So which slope stability is very important, especially these days, in which we have forecast prediction that bad rain would be pouring down in some part of Italy. Professor Paolo Dorico is coming from University of Berkeley. I said University of Berkeley, no, the university nearby. I'm from University of Verona, so very welcome from University of Berkeley, and it's a Department of Environmental Science. And Riccardo was promoting the ousting Paolo, and very happy to introduce him as a global water resources and natural resources according to global change scenario. So again, we have in the foundation and in the University of Trento people working in global change scenario, both at the forest level and ecosystem level. So I think it's very interesting to discuss and to listen to your talk. And then we will have Jill Rosenthal, Professor Jill Rosenthal at the University of Padua, from the Department of Biology, and his research is on evolution of mating decision in organisms from protozoan to humans, animal behavior. We are talking animal behavior, which is a very high topic in this moment for Trentino. So this is something that we would like to be distracted sometimes to have discussion, also scientific discussion, not political discussion, but scientific discussion about those topics that are so important. So genetics, hydrology and environmental changes, and climate changes, and animal behavior and zoologies. So that would be the topic, and then there would be the community of the PhD students that would have the roundtables to discuss. So I would give the floor to Rico first, Professor Rico Pert, that he would present his talk. And thank you very much for accepting our invitation. And please, Rico. Full screen.
Preferisco il gelato. Okay. So, good morning, everybody. You know, normally when you go to the congresses, you listen the litany, oh, I'm very happy to be here. I wish to thank the organizer for inviting me, etc. I always hate this. But in this particular case, I am really happy to be here. Mario before said, Enrico is my friend, even if he's a genetist. This is what was the meaning of. So what I'm going to tell you is a little bit of a part of a story that started 2010, around 2010, when I was at Scuola Sant'Anna. I was engaged in a PhD program, which was specifically devoted to students coming from the so-called emerging countries. And so we started to reconsider the type of PhD activity that we could have proposed to this type of students. And then being a geneticist, I started getting interested in studying agrobiodiversity. In fact, the PhD program name is agrobiodiversity. And so we started to interact with students coming from Ethiopia, and that was the starting point of this part of the story that I'm going to tell you today. And giving part of the story, I will also show you what we learned along the way. And I think that some of the lessons that we have learned might be relevant also for you. So the title is missing the first slide. Yes. So in the title, there are all the keywords that you will listen during all my presentation. So genomics is the first keyword, but next to genomics is traditional knowledge. So the effort, so that you, the line that I'm trying to trace here is how can we combine advanced research, advanced technology with the traditional knowledge. In this particular case, the traditional knowledge of farmers' community. The other keyword is agrobiodiversity, and you will see why it is important biodiversity. And finally, Ethiopia as an example. What I'm going to tell you is mostly the results of the activity that started in 2010 in Ethiopia, but now the lesson and the methodology and the models that we developed are also trying to be adopted and transferred to also different contexts in Africa. So, the topic is agriculture of the 21st century. So here in the two pictures, you see two extreme model of agriculture. No? The agriculture that we are used here in our environment, highly industrialized. But next to this, there is another extreme, which is the so-called agriculture for sustaining the livelihood of the people. And the two agriculture, obviously, you know very well, are very, very different. No? In the first case, we have homogenized environment, a lot of inputs, utilization of improved material, genetic material, and so on. On the other hand, the so-called smallholder farming, because this type of agriculture mostly is done by farmers' community which have a very small plots. We are talking about plots smaller than 1.5 hectares. And it is characterized by extremely variable agroecologies. And this immediately tells you the difficulties of identifying the proper genetic material which is best suitable for the different conditions. Low, if not zero input, mostly rain-fed, poor access to improved material, and unfortunately, a lot of this type of agriculture is also in context of highly political instability. Ethiopia, which seemed to us a very stable country, you know what happened in 2021 with the terrible civil war. And then, obviously, exposure to climate change in a fragile context. However, not everything is a negative, because they are rich very much of agrobiodiversity. Obviously, I don't want to give you as a wrong impression. Not all agriculture in Africa is of this type of smallholder farmers. There is also highly advanced and so-called industrial agriculture also in Africa. But smallholder farming is very much distributed in all the African continent, especially in marginal area, or at least defined marginal compared to the industrial type of agriculture. And dozens of millions of people are involved in this type of agriculture. Okay, so we geneticists are a little bit arrogant because we believe that genetics is the center of life science. I believe that, Mario probably too. And so, being the center of life science, we also believe that we can contribute very much to the various problems. The two types of agriculture are very different, but the major problem, the major challenges that we have to face in this century are exactly the same. So what could be the contribution of genetics for 21st century agriculture? Well, first of all is, I put in quote, produce better varieties, better material to be grown. But better is obviously very depending on the situation. So define what is better is not so trivial. And yield potential is one of target, but not only. Besides the potential, we also need to have stability, so response to constraint, so tolerance or resistance to stresses and so on, but also to produce products that are valuable for transformation, for utilization, but also of good quality and efficient to utilize the resources. So what are the tools that the geneticist and the plant breeder can utilize? Well, there are two pillars. One is the genetic variability. When I teach the basic course of genetics, there is this mantra, no genetic variation, no genetics at all. So once in a while I say, what is the mantra today? No genetic variation, no genetics. So in order to produce better varieties, you need to have genetic variation on which to work. And the other one, obviously, is the plant breeding. I'm very fond of this picture, which depicts a field in an international center in India. It is a sorghum field, and is one of the many dozens that I saw in this international center. If you can see, each plant is covered, and it means that the reproduction of each plant is controlled. So you can imagine the amount of energy, amount of work, amount of time and effort that is behind the plant breeding. Therefore, everything that can speed up, increase the efficiency of plant breeding is more than welcome. A little bit note of color. In the morning, in this field of sorghum, they used to send, I don't know if they do this still now, they send women clapping hands and making noise to scare the snakes, to scare the cobra. And so this, I never entered into this field, to be honest. Okay, so now we have the two pillars. So you probably know very well that the genomic revolution has changed very much the work in life sciences. And the results for the geneticist is the fact that we have incredible capacity to identify genetic variation. So identifying, assessing the type and amount of genetic variability is no longer the bottleneck. The bottleneck, so for instance here, just a simple variation in a sequence, identify a variation. So what is the bottleneck now is to give a meaning to the variation that we are able to identify. So now, so the technology is way changing very fast. We might want to discuss about this if you want. So, for instance, let's suppose that you have characterized the sequence of a number of different individuals. So each row is sequence of an individual. Each column is the same position of the same sequence. So you can compare the sequencing data and you can immediately identify the genetic variation. So genetic variation no longer a problem. On the other hand, if you have genetic diversity, genetic variability, then you can try to combine the information coming from the genomic analysis with the characteristics you measure with in your gene pool, and then try to identify which part of the genetic variation that you measure is relevant for specific traits, for specific characteristics. So another point that maybe is obvious, but is always better to underline, is that the genomics brought life science into the big data scenario. So genomics can be seen as a digitalization of the genetic information. So, okay, so the challenge, as I said, is to assign a function to the DNA sequence. So the other keywords was Ethiopia. So why we started working with Ethiopia? So keeping in mind what I told you about the necessity of having genetic variation, Ethiopia has been defined as one of the Vavilov center of crop diversity. I don't know if you are familiar with the figure of Vavilov. Vavilov was a botanist, a Russian botanist. He was a pioneer in identifying area in which there was a lot of genetic variation linked to crops. And so he identifies regions which were potential zone of origin of domestication, for instance. The story of Vavilov is also very sad because he entered into conflict with Stalin and he finished his life in a prison or a concentration camp. So Vavilov is a big figure for all of us, all our researchers that study genetic diversity, especially in plants. So as you can see, I hope that most of you know where Ethiopia is, here on the Horn of Africa. And I put the name of some of the crops that have been domesticated in current Ethiopia or secondary area of variation. So, and I sign in specific coffee. Coffee was domesticated. There is a nice story about a mythical story about the domestication of coffee. If you want, I can tell you later at the coffee break. In red, I highlighted the crops on which we are currently studying. So barley, durum wheat. Durum wheat was the starting point, and the rest of my presentation deals with the durum wheat. But also teff. Here you see teff is this field. Teff is the most important for Ethiopia and Eritrea, is a staple food, is the most important cereals. And probably you heard about the importance of teff, which is gaining importance also in the Western world as a superfood for its characteristics. Why is also interesting to work in Ethiopia is because a lot of the variation is still maintained in the field by the farmers community. So what we call the land races now, the terms has been changed and people prefer to call them farmers variety. So they are at the interface of natural selection and anthropic selection. And besides having maintaining the field a large quantity of genetic variability, there are also in ex situ gene bank, and in Addis Ababa there is the biggest gene bank in Africa, where you can collect many accession of the different crops. Okay, so what is the first step if you want to start to characterize the genetic variation available in any area? Is you have to assemble, to collect a number of different potential genotypes, different potential varieties. And in jargon, we call it a diversity panel because it's a panel of different varieties of the same species, which can be representative of a part of the variation of a large part of the variation. So if here you see the picture of Ethiopia, and the black dots are the region where specific accession has been collected and either maintained in the gene bank or are still cultivated in the field. So as you can see, you try to cover as much as possible the area of cultivation, in this case of wheat, and you can see also in the different color also the orography and the geographic characteristic of Ethiopia. And in fact, in Ethiopia, you go from lowland, what in Ethiopia is considered lowland is 1,000 meter high, up to highland, which is above 4,000 meters. Yesterday, I was talking with Mario. Mario was surprised when I told him that you can see barley fields up to 4,000 meters high. So you collect, it is important when you collect the material to have as much information as possible. So what they are called passport data. So when they were collected, where they were collected, what type of characteristics. Sometimes you can always trace back the family of the community which provided the material. So what you do once you have produced this diversity panel, you do the genomics. So you extract the DNA and you characterize by sequencing analysis the structure of each accession, and then you compare. So you produce that matrix of variability data. Then you have to interpret the information. One of the way, for instance, is to utilize this statistic, which is called principal component analysis. I don't want to enter into the specific of the analysis, but in very simple terms, this methodology maximizes the differences between accession. So here you have two principal components. Each dot summarize the genomic data from that specific accession and is put into relation with the other one. So in our diversity panel, we had a large number of Ethiopian accession, farmers variety in green, but also we had a subset of material coming from the Mediterranean area, and also all the improved durum wheat variety that has been approved for cultivation in Ethiopia. And what you can see, so this summarize almost more than 30 million data points. So you can just see how much, why I told you before the genomics brought the genetic analysis into the big data scenario. So what you can see is quite evident. So Ethiopian farmers variety are very diversified. So they are spread along the principal component 2. And they are clearly separated by the, from the Mediterranean material and also from the Ethiopian improved variety. And if you look, both Ethiopian improved varieties and Mediterranean material are clustered. So, meaning that the genetic variation is much reduced as compared to the material that we analyze as farmers' data. Obviously, there is a bias because the number of farmers' variety was higher than the number of Mediterranean material, but still it's quite clear. And this, the last point that I underline here, is crucial. So it seems, no, there is quite a strong evidence that the original Ethiopian germ plant material has not been utilized in breeding, not even for the breeding to cultivate it in Ethiopia. So this was, keep this in mind. So how can you utilize this type of information? Well, in our case, we were interested in say, well, if the Ethiopian agrobiodiversity has not been utilized, why don't we try to grazie mille. Thank you. Questo è il centro. Okay. So, see the difference? So, the idea is, why don't we start to mobilize the genetic variation that is contained into the genetic material? So, what is the tool? The tool is to do breeding, to do crosses. And in fact, what we decided to do was to produce a population, new population of durum wheat, in which we cross a number of accession, farmers' accession, with an improved variety. An improved variety which was highly utilized. So, this is the improved variety which has been crossed with all these 50 different accessions. And from each of the cross, you derive by inbreeding, so by selfing plant for a number of generations, and you end up with a number of, in this case, 50 families, each family derived from the original cross. So what is the advantage of having this type of genetic structure? Is that you distribute the combination blocks in this genetic material. And the different families are interconnected because they contain one of the parents. So we produce a high number of lines which are highly homozygous, meaning that there is no internal variation for each of the lines. Then we wanted to see what type of structure this population have. But what was the objective? Yes, to put into the scenario new genetic variation that has not been utilized before. This was the first objective. So to produce a platform for but on the other hand, we selected the parents also for interesting agronomic traits in such a way that eventually this high number of lines could contain some interesting combination which could be or immediately utilized and distributed to farmers, or could be the basis for further breeding. So this is, before I show you the principal component, which is one of the way to describe the variability. Another way is to produce what is called a tree. And as you can see, each line represents one of the accession, and as you can see, the elite varieties and the Mediterranean varieties are branches out, so meaning that the genetic variation is high. So the colored circle represents the 50 accession that has been utilized for producing the population, and as you can see, we try to cover, to have representation of all the majority of the variability that we observe. Then we did not have the manpower and the resources to characterize all the 6,000. So we decided to characterize 12 families, 12 out of the 50, 100 lines for each of the texts. And this is 10 minuti. Thank you very much. I will go fast, but here is another way of seeing. And as you can see, the different colors represent the different families, and they are separated. So we were very happy to see that the job was done. And then we make the principal component to see whether we close the gap between the elite varieties, which are here, the founding varieties, and these are the material that are used. So we effectively try to close the gap and put available new genetic material. But what can you do more? So on one hand, you have the matrix. But if you have also done an agronomic and phenotypic characterization of the same accession, you have a second matrix. So here you have the matrix of the genomic data. Here you have the matrix of the trait that you have measured. Put together, you do advanced statistical analysis, and eventually you could identify region on the chromosome which affect the variation of the trait of interest. This is the name of this type of graphic is Manhattan plot, and it is a statistical estimation of the link between genetic variation. Here you have the chromosome. Each dot represents one genetic variance that you can observe. And here you have a threshold, a statistical threshold, and then you can decide which of the signal you want to analyze. Okay. Now, in the last 5 minutes, I have to go to the other players, which are the farmers. So, exploring farmers. So I told you already that this type of agriculture is characterized by small plots. They use mostly their own seeds. They don't have access. So the problem, and this might be a little bit cynical, but these farmers, the well-being of their family depends on the capability of producing and obtaining products during the season. So they must be very efficient. And the efficiency comes from the experience, comes from their knowledge. So knowledge is something that at this point should be or could be utilized. That was the idea. So the provoking thought was, there is this knowledge, which has been selected during centuries and transfer from generation. And so one thing is, but are we sure that this type of agriculture is really marginal? So maybe it's marginal from our point of view, but it's not marginal for them. And can these smallholder communities be a resource instead of being considered? And eventually we come to the original idea that I expressed before. No? Can we join modern technology with tradition? So in order to do this, you have to devise what we are called participatory approaches. So we did not invent participatory approaches, but we modified the way of doing participatory approach, which was to introduce the genetic approach. And instead of asking the farmer, do you like this or you don't like this, select among the 200 varieties which one you like. But we change it. Well, we transform the evaluation of the farmers in numbers. So we ask the farmers to look at the plot without knowing what was there, analyze traits, and give a numeric evaluation. So each farmer independently gave an evaluation from 1, I don't like it at all, 5, I like it better. So we collected a number of data points. But the question more theoretically, but what is the value of this type of participatory approach? No? So I have to put the eyeglasses, otherwise I don't see. So the first question is farmers' knowledge, can we consider the evaluation of the farmer as a trait? If yes, we have to demonstrate that the evaluation is consistent, that is repeatable, and is heritable. So it does not change from one generation to the next. And then, what is the relation from the farmer's evaluation with the traits which are measured by the breeder? And finally, if this is true, can we identify, this is a big intellectual jump, can we identify the genetic basis that can explain the farmer's choice? And here I just show you how does it work. So on one hand, you have the diversity panel or the new population, the breeding population that you have used, and you genetically characterize them, and then you also test it in the field. So you eventually produce a number of data points based on metric education. On the other hand, you can engage the farmers' community. And in this case, you utilize farmers, men and women, who are really working and are experts, in our case, of wheat. So they were wheat growers. The first step is to meet the farmers, discuss with them, do what are called focus groups, which they explain what they grow, what are the limit of their production, what are the expectations, what type of material they would prefer. And eventually you come up with some priority traits which you can ask the farmers to analyze directly in the field. And then you do work. So divide the farmers in groups, homogeneous groups, different genders, so you don't want to have any influence. And then Then you ask individually the farmers what is the evaluation. So look, you are in front of this plot. We are looking at earliness, like the length of life cycle. Do you like it or not? If you don't like it, 1. If you like it a lot, 5, and so on. And finally, so they go across the field. So you imagine that we had almost 400 plots multiplied by 2, multiplied by 2 locations. So the 2 locations had the 2 farmers unity. But they spent quite a number of days together with us. So it was really also a very touching. And this is the number of data that you can produce by this. So 8,000 data points by the researcher, close to 200,000 data points by the farmer. So it's a huge number. But if you remember the scheme that I show you, here you have the matrix of the method frame. But also change and utilize farmers. The approach is exactly the same. But in this case, you might be able to identify chromosomal region which justify some of the farmers. Just to complete, this scheme is relatively simple and can be adapted to a number of different types of traits. For instance, I show you in the map the position where the accession were collected. So we had information of the position. We could derive climatic data. So you could also have bioclimatic data. So again, you have the matrix. So you utilize the same genomic data on different matrices. Then you combine all this. And I see here. And what are the outcomes? Yes, the more farmers provide quantitative and reproducible data. Farmers' appreciation can be considered a quantitative trait. And this is the big result for us was the fact that yes, we confirm that traditional knowledge can be utilized for and can be included in modern breeding, especially for situations like the one that I described before. Finally, obviously, the professor talked, but the work is done by the PhD students and by the researchers. So here are some of the people that I want to acknowledge, all PhD students from Ethiopia, from Eritrea, postdoc, Carlo Farda, researcher at Fiat, and who was the fundamental for us to introduce the socio-economic aspects of our research. Because obviously, when you start to interact with the farmers community, you cannot just talk about the natives. You have to consider the context. Obviously, the farmers, but also the driving force, which is material. So, thank you very much.
Thank you, Enrico. I think we have time for one.
Yes. Well, the statistical analysis is basically the one that you use for GWAS and for QTL analysis. So linear models and also Bayesian models. Both, yes. And also depends also on the structure of the population that you analyze. if it is a structured population like the one that we produce by breeding, or is a diversity pattern. Oh.
I am researcher. I was curious about if you had any results with the association with the farmer score.
The
If you have-
association with
Yeah,
between
if you have-
farmer score and
yeah.
metric traits.
Yeah, if you found any association candidate.
There is correlation. So some of the of what you find. Okay. There is correlation between farmer score and some of the traits. What is more interesting to us is once you do the mapping, what you see is that sometimes you identify same region. In other case, matrix analysis identifies specific loci and not the farmers, and vice versa. So you can interpret the farmer score as introducing an additional layer. So they introduce time because they base their evaluation on their experience. Yeah. No, very good, very important aspect. So the first answer is all the work that has been done, especially on the genetic material, was done in Ethiopia. We never took out a single seed from it. So we could provide the genomic analysis in Pisa, but the analysis was done engaging the PhD students, but material was not brought to us. As for, yes, these are data that are available. And so, in principle, everybody can utilize. Obviously, the difference between the farmer's contribution and a normal breeding strategy is that normally breeding is done in many locations, and the objective is to produce a limited number of variety which can cover different agroecologies. Here, the focus is different. You want to identify the proper material for specific location, and in fact, the ideotype, so the type of plants that the farmers prefer, sometimes is totally different from the material which is produced by the breeder, by the breeding. And this is a big problem because I told you before that there is not much access to new material. And so there is also a problem of acceptability by the farmers community. For instance, very productive durum wheat varieties are short with a lot of energy put into the spike, the seed. But the farmers have animals, so they need the straw. They need the straw to hatch houses. They need the straw to mix with the mud to produce the house. So the idiotype is totally different. And it is not of much interest to, I believe, to Monsanto. No, but could be. Why not? I don't see a contradiction because obviously the effort is for the community. But these are genetic resources that can be useful for all of us, not only for them. So in principle, I don't see any big contradiction, but it should be balanced.
I'm a researcher. Thank you very much for this very interesting talk. I'm a ecologist, so it's maybe, you know, my questions or my observation might be very trivial, but I was really fascinated by this combination between what I understand it's basically phenotypes, understanding of the phenotype, that it's really built up by a lot of experience by the farmers with the highest genomics capabilities. It's really fascinating to put this together. And I mean, this happens a lot in ecology too, where sometimes you have to be quick in understanding the patterns without really knowing the process, which is the genetics and genomics after all. And so my question is about the resilience, right? I was very impressed by this point that they really need to build a knowledge. So in essence, it's not for us maybe the matter to have a more productive production of food, so maybe to earn more money or to be more effective. In this case, it's more really survival, right? So, so what did you learn about resilience, for example, to changes, to, you know, including climate change?
Resilience is eventually the big target. But as I started to say when I answered to the previous question, is that here the effort, I could give another hour, another seminar with a different angle, maybe next year, if my friend Mario will invite me.
Despite you being a geneticist.
Yes. Okay, is to identify specific material for specific conditions. So depending on the condition and depending on the we touch on only the surface. So what we are trying to do now is to increase the number of farmers engaged in the evaluation. But eventually the evaluation is the results of the both analyses should brought to the identification of a number of accessions which are selected both because are contain specific genomic structure and are also appreciated by the farmer.
Thank you.
Thank you very much. First, we are already late.
Sorry.
No, but it was not your fault. We started late. We started late, so we are keeping the lateness, and but we will start with the first roundtable. So, who are the person involved?
Okay, we start with the first round table of agrosystem area, and I ask Professor Riccardo Rigon of the University of Trento and the students that I'm going to call now, coming to the stage, Sara Vesani, Gelal Fouani, Giorgio Licciardiello, Stefano Nadalini, Roselli Gerardo, Scala Marica, Francesca Cutrupi, and Jim Yates that should be connected now. So I would like to open Zoom.
How many minutes do we have?
Yeah, that's good.
Okay.
So now we are waiting. Jim, the PhD student, is going to connect again. So the roundtable aims to give the opportunity to PhD students to exchange ideas, to discuss with a senior researcher about topics that are related to their research. So for this aim, I would like to ask students to present themselves with a very, very quick self-presentation with name and the topic of their research. Thank you.
If I can add, in your presentation, you should also, in just few words, say what is important in your research. What is the most important thing that you have done for the scientific community and the science? Okay. Words.
Good morning, I am Sara Vesani. I'm working with Pinot Noir that is a susceptible grape wine genotypes, and I treated the Pinot Noir with volatile organic compounds identified in resistant grape wine genotypes. So we verified the reduction of downy mildew symptoms and the upregulation of defense-related genes and accumulation of defense molecules in grape wine leaves. So it's important in my project to have identified the activation of defense mechanism in the grape wine leaves with application of molecules produced by resistant grape genotypes.
Hello, everybody. My name is Giorgio Licciardello, and my supervisor is Michele Perazzolli. And my PhD aims at the determination and characterization of the metabolome of tomato plants, which are bacterized with cyclotolerant bacteria. For those who are not familiar with this word, cyclotolerant means adapted to cold. And the aim of this research is to determine whether to study natural conditions, to study the nature, so the interaction between the bacteria and the tomato plant, in order to discover the metabolites that may help in the acquisition of resistance to cold stress.
Good morning, everyone. My name is Jalal Fouani. The main topic that I was studying is regarding a very economically important insect pest, which is Allomorpha alis, or the brown marmorated sting bug. The importance of my work is that, and with the help of the group which I'm affiliated to, is that we try to study an important ecological trait of this pest, because they communicate via substrate-borne vibrations, and we try to exploit this trait in a more applicative way for further IPM, integrated pest management strategies for more sustainability in its control.
Good morning, everyone. I'm Marika Scala. I'm a PhD candidate. I'm about to submit my thesis. And the topic of my PhD was the study of invasive insect pests and the biological control mainly. For this reason, I studied the interaction between this pest, this insect invasive pest, and their sympatric parasitoid, for example, Allomorpha alis and egg parasitoid Cystoscus mitsukuri, but also the fly Drosophila suzuki and its larval parasitoid Ganaspis brasiliensis. Moreover, I characterized the vibrational communication that is involved in the mating behavior of the invasive pest Bagradelaris.
Good morning to everyone. I'm Stefano Nadalini, a third-year PhD student, and I'm studying the downy mildew of grapevine, so Plasmopara viticola. And in a broad aim, I'm trying to provide new tools for integrated pest management strategies. With, on one side, the description and the management of fungicide resistance, which is quite a hot topic in our field. And on the other hand, in collaboration with a company, we are developing and characterizing 3 new candidates to present them as new fungicides on the market based on plant extracts and biocontrol agents. So I guess from this point of view, the most important aspect will be if the company still has the interest to propose 3 promising candidates on the market and having them characterized, which is quite an interesting aspect for farmers at this point.
Good morning, everyone. I'm Gerardo Rosselli. The focus of my PhD program is to develop a stellar insect technique for the management of Allomorpha alis. So this technique is mainly based on mass rearing irradiation and release of sterile males. And those males should be able to compete in the field of wild males for mating. So, however, for such a species is considered impossible, the mass breeding. So that's why we focused on development of an alternative tactic that involves the wild mass harvesting and then irradiation and release of those insects. And then we focused also on the development of the combination of sterile insect technique and biological control, and we evaluated sterile eggs oviposition substrate for the exotic ecparasitoid Trissolcus japonicus that is considered the main candidate for the biological control of the species.
Thank you. Good morning. My name is Francesca Cutrupi. During my PhD, we detect SARS-CoV-2 in wastewater. And the main application was the strategy or WBE, wastewater-based epidemiology, to give this approach that is based on the detection of the concentration of the virus in this savage, in the wastewater, to give to obtain information on the health status of the community. Specifically now, our work help the health authority to make a monitoring, surveillance of the SARS-CoV-2 case, because now the swab test, the rate of the swab test decrease a lot.
Okay, Jim, is your turn.
Everybody, hi, Professor Rigon. Sorry you have to see me in a big screen like that, everyone. My name is Jim Yates, and I'm a third-year PhD candidate. Can you hear me? Yep. Yes, the focus of my research is on forest monitoring and ecology, and we develop IoT-based platforms for detecting several different ecophysiological functions for tree behaviour. Specifically, I've been building dendrometers, actually, to detect radial increment change at individual trees are a really cost-effective solution. And from my contribution to the research, we are detecting, for example, in one research site, the dominance or the behavioural dominance of Quercus charis sprouts on coppice forests, which is important for silvicultural intervention practices going forward. And I've also got research sites in mountain gradients looking at the way that forests respond to different climate stress indicators in the Apennines.
Okay. So what's next? And most of you are working on plant defense, let's say, from different points of view. Did you have the, now I am doing the question, but you can do questions to each other also. So when I am giving you the words, please do a question to at least one of your fellow. And my question is, what was missing from the from your PhD studies here? How was the interaction between you? And what you suggest to the new coordinator of our PhD students to improve your experience and your research path? So we start from the last one, maybe Jim first, and then we go back to the others.
Well, my experience obviously dictated by two different factors. The fact is that I'm actually based in Lazio under Riccardo Valentini. So it's not only that, I decided to have a child. So it's very difficult for me to come to FEM very often. So I haven't had much interaction with the other PhD students, which is really circumstantial rather than anything else. But the times that I have been at Mark, it's been absolutely wonderful and accommodating.
Do you have questions for your fellow here?
Actually, I was interested in, if I understood correctly, it's not my field, but this detection of the vibrations between the insects. Third speaker, I'm just wondering how do you detect this frequency oscillation if you use a specific type of sensor, or maybe I missed something in the actual presentation. For the grouse beetle, I think it was, possibly.
Let's go in order. Then you keep the answer for when you arrive at your turn. So otherwise, we go to interaction grows more than exponential. So, so Francesca, you were the last one. So.
I think that for us that we started the PhD in the 2019, the problem was also the COVID experience, so the problem of the pandemic. I think that for sure I didn't understand so well my colleague Jim. I don't know if I repeat what he already said, but I think that this topic is this PhD course is great because there are a lot of different topics, but it's also a bit dispersive. I don't know how to say. I would like to suggest more a situation of meeting, because for me that I am also an environmental biologist, could be really interesting know more about the work in agrofield of my colleague, but there was a lack of this type of meeting. And I
take note.
I imagine that is also difficult because we are also so deep in the research, and also it could be difficult to organize this type of moment. I have a question for Gerardo. Because I know something about your topic. If you could explain me more about the work that you carried out.
So, who was just before her? Yeah, you were. Okay, that's good. So you can answer directly.
Okay, so first of all, thank you for the
please
question.
speak aloud. I am a little becoming old, so.
First of all, thank you for the question. Okay. So, first of all, I will answer your question. So, what we did was, first of all, to develop effective live traps in order to have mass trapping. Then we focused on the finding of the right dose of irradiation, and we focused also on the mating behavior studies of the irradiated insects. We got an agreement with the Santa Chiara Hospital here in Trento in order to improve the irradiation times and the quality of the insects. And then we evaluated also the suitability of sterile eggs, considering the possibility of release insects in the spring. Of course, a lot of things need to be done. So it's early to say we are ready to release insects. We are still in the laboratory phase, but there are some bases for the improvement of this technique. So.
Okay.
About my experience here,
yeah.
I think it was biased by the COVID, as Francesca said, and of course at the beginning was not the best situation for us. I renounced to my period abroad, and I chose to spend more time here in other collaborations. Fortunately, I was involved in other cooperation with other researchers.
Well, you look
okay.
at them.
Okay. Sorry. So that's it.
Who is next?
So about my experience, I will almost totally agree with what Francesca said, even though I started my PhD program 1 year later, so in 2020. My experience was still biased by COVID. So, for example, the first month we weren't allowed to meet. So these experiences we are facing now were supposed to happen, obviously for health reason. But even exchanges between us, we were remotely, so we didn't have the chance to even speak among each others and to have, let's say, a confrontation from the social, but then on the scientific point of view. And for my point of view, this was the biggest issue, let's say, at least at the beginning. For about the question, I will ask the guys who are working on integrated pest management on the insects part. If you had some issues or if you know the perception about the techniques you are developing or studying from the farmer's point of view. I mean, from my point of view, new products, let's say they have been weighted like gold, let's say, since we have less fungicides, less tools, less efficient tools to, let's say, control diseases. But on the other hand, farmers tend to look for the 100% efficacy, the magic product, which is able to defend the field. And so new products which have a lower efficacy, let's say, has been seen like in a skeptic way. So is that the same experience you had, or is it just from our side?
Okay. So next one.
So I will answer your question first. And you know, in IPM, that is integrated pest management, you have to think about different strategies combined in order to reach the goal that is keep the pest, the disease below the threshold. So it is impossible to reach the 100% of control. I think this is really impossible way to think about, and I know that the growers won't reach this goal, but I believe that in most of the cases it is impossible, especially if you're working in open field, while if you have a semi-field condition or a greenhouse, maybe it's easier, but I don't know. Regarding the first question, I can say that I belong to the generation that experienced all the restriction of the COVID. So as Francesca and Stefano, I think that maybe the best case is have more meeting with each other from different fields in order to, I don't know, discuss about different topic, and that's it.
Good.
Regarding the first question, I don't want to repeat what everyone said. It's basically they covered all aspects of the whole experience that we had to go through. Regarding, there were a couple of questions regarding yours first. Just to add something to the point of view of Marika. Like she said, it's impossible to eradicate something unless, I don't know, with the sterile insect technique perhaps, in some certain environments, like an island or very controlled environments, you can possibly eradicate some invasive species, but this is very hard and very unlikely in most of the cases. So I just wanted to add that. Regarding Jim's question, you asked about which kind of materials we use to record the vibrations and the substrate-borne vibrations of the insects we study. So there's a multitude of materials depending on your research question and on the environment you want to study the insects in. For instance, you have the very technological lasers that work on the Doppler theorem. So they measure the small vibrations of the substrate, and that's how you get an idea of their communication. But these are usually super expensive and very hard to carry around, but they are very sensitive, obviously. So they are the best material to use. However, if you want to do something in field conditions, you cannot carry on 5 kilograms per laser around, and the laser beam, if there's a bit of wind, the laser beam will not always be focused on the substrate, so you will lose a lot of data that way. So there are different kinds of materials that you can use, like transducers and small accelerometers that they are very lightweight. You can carry around anywhere, and you can just put them on the surface. But obviously, they're less sensitive in comparison.
Thank you, Jalal.
Last but not least.
No, it's not the last one. This.
Yeah, you are so okay. Sorry.
Yeah, so By the way, of course, I do pile up on the general complaint about the meeting aspect of especially the COVID times. But yes, of course, now let's say the restrictions are over. I simply pile up on other people observations, and I want also to tell that not only scientific meeting like this. I come from an entire year in Vienna where also social activities were organized at all the levels, not only the single unit or group, but an institute level. And so maybe you start with no scientific purpose at all, and then end up, of course, communicating and knowing people and that sort of stuff. Yeah. Regarding the question, I will simply ask Sarah if she could. Okay, I know a little bit of your project, and I wanted to know whether you identified the metabolites involved in your biological question, and if they more or less belong to a few or a single category of metabolites like polar compounds or polar compounds, or do you or can you see like isolated metabolites from different categories, like amino acids, one amino acid, and this kind of test?
Thank you, Giorgio, for the
Sarah,
question.
please.
So, okay. Also, for me, at the beginning of the PhD, was difficult to know other colleagues for the COVID situation. So, okay. Also, for me, at the beginning of the PhD was la voce. Okay. Was difficult to know the other colleagues caused by the COVID situation, and sorry. Another difficulties was to frequent the online courses, and I can suggest for the next years to make these courses in a non-frontal way, but to give to other students the opportunities to know each other during, for example, working group. And to answer to Giorgio, I identify metabolites in different chemical classes. So the first I searching for the chemical classes, and for the untargeted metabolomics that I use as approach, I use a techniques for
Loader.
medium polar molecules.
If there is some question from the audience.
I would like to comment the meetings and the possibility to have a more moment of interaction for the PhD community. I would like to launch the idea to have the seminars that there comes. I'm proposing of seminars, internal seminar for the FEM, or together with the C3A, whatever, for the entire community, and the decided an annual schedule, and have 5 seminars every year, so to have the contact, and this could become from a huge interaction of disruptive and seminar talk from guests from all over the world. I can put some money in these ideas together with the University of Trento, but I can put some money for doing this, the possibility of doing these seminars that are scheduled by you, directed by you. So this is for the entire PhD community.
I take the offer, and I give to the new coordinator of I think it is a great idea, and so we will develop the thing. So we also praise the general director of the FEM maybe to provide some wine when we do that. We would have conversations. There are other questions from the audience. Some question that you want to ask to the students. Otherwise, I think we can. Okay, okay.
I'm sorry, I will use this opportunity for asking to my colleague, but also to the researcher with more experience than mine. I had, I don't know, to say not a problem, but during my PhD, the fact that the thematic, the COVID situation are changing continuously was give us as a researcher a rush to change topic, to understood what was the focus of our research continuously. We found this situation really stressing. I wanted to ask if someone else has this type of experience, that you are working, but the research around you are faster than you in your work, and how to, if there is like a strategy or an approach to use in this kind of situation. I know that could be a particular situation because the pandemic was something really specific. But I think that this rush in research is something that perhaps also other people could feel. And
I think, Francesca, you just described research in a nutshell, because everyone is in a rush to publish. There's a lot of competitiveness, at least in our case, for instance, with the new insects, that everyone just rushed to discover which are the ways to control under a certain threshold and so on. And you have, I don't know, at least 10 labs on the Italian level that are working on the same thing. So you're always, I want to do this thing if I don't need to take a break, for instance, now. Otherwise, I can lose an important research finding and possibly a publication. So yeah, I think it's probably in your case, it was much crazier because you worked on a topic that on a worldwide level, everyone was working on it and just publishing new stuff daily. But I think it's quite normal now, and there's this huge how to call it, pressure to publish, and like the more you get publications, the better you get your name out there.
I have to say that to keep the rate of publication of Francesca is quite demanding, but
it's a very interesting point. During the COVID time, I had a very great idea as a behavioral ecologist to study COVID-19 lockdown effects on wildlife. So when I had maybe the time to do, like to finish off some stuff, I started a new initiative that blew up with 300 institutes, 1 billion locations in Europe, and it was like a total nightmare. We rushed to publish in Nature, and so it was a total nightmare. I mean, exciting, but it was really like the case where you take an opportunity. So I think this is really a little bit in the research mode, to take opportunities and to really be uneasy if you haven't taken them. So no one really tells you that you must do it. I mean, sure, in the PhD, you have your chapters, you have to finish, but even when you go on, there's not really, I mean, it's not that Mario calls me every day, says, so did you work on this analysis? Did you take this opportunity? It's really your internal rush. And I think it's the word, but it's really also personal balance that one has to seek somehow. And what I try to think myself is that it's important to take the opportunities, but it's also good. Maybe at a PhD level it's difficult because you have to have your stack of publications to make a career. But when you go on, to think ahead and also reflect not only on the current opportunities, but also on what things are going to count in 5 or 10 years. So what kind of analysis, what kind of papers are going to be still important in 10 years' time? And it's a balance between the two, like rushing to catch an opportunity, but also try to stabilize some knowledge. That's my own take. It's a personal one.
Not that Paolo is saying what I wanted to say. I don't know. No, I realize it's a student forum, but the same issues come up also in my university, and so I want to offer a couple of thoughts. First of all, when I started my career, I was hired and I was in a foreign country. I never been a student in America, so I always felt like the one who was getting through the back door, and I will never be good enough. And there is sometimes, and you compare yourself, and then overall we are all the same. It's not that it's not the paper more that your colleague publishes that makes the quality of research better. And besides, some of the people who were always coming from this Ivy League, etc., were not. We were all the same. I want to say so. Never. I think your generation is facing more problems, more challenges, and I think it's mostly related to social media. Because this is the modern culture of displaying what people do, which in our world, I didn't know what my colleague in Florence was doing or other things. So it was a little bit less of, maybe I'm not going to oversimplify or saying that you're dealing with a world that is structured in a different way. So perhaps that plays a role. But I agree with the fact that overall, of course, by the end of the PhD, you need to have a certain number of publications to be competitive, blah, 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 etc. But sometimes it's not only the number, but it's the quality, the ability to work independently, the ability to present your work well. So there are many factors that play a role. Most importantly is the type of research, how new, how interesting, how relevant. So it's what I'm trying to say is that there are different metrics that is not only counting. But I realize that I think social media play a very important role because there is too much of comparison and sometimes. We go for hours if we want here.
Francesca opened a very interesting topic. Okay, I'm a PhD candidate and a researcher here at the Foundation at Denmark. So I'm a colleague of Francesca. And in my case, Francesca had a particular condition because she studied, like the other colleague, exactly COVID. So I think her condition is very different from the other ones. I had the opposite condition where I was completely blocked. So I couldn't do anything for at least 2 months during lockdown. After a while, I could go back to lab activities. And so for me, COVID was like a stop. So I perceived this rush, this for your case. For me, it was just not relaxed, but what's happening, I don't know. I want to introduce a new stuff because actually you talked about the problem of publication and competition that is normal, as the other said, but was more urgent during COVID lockdown. But we have, as highly educated people, we have another role in the society, and I think we partially missed it during the pandemic. In particular, in communication, we have to keep in mind that we are dealing with numbers, we are dealing with statistics, and we know more or less what we are talking about, but the other people not. And I think the scientific methodology is not the basics of everyone at luggage, personal luggage. So when you say, you in general, when a scientist say on TV or everywhere just a number that is a percentage or an absolute number, you are giving a different information. So not the same information, but it has a different impact on the effect this information is understood and considered by the people around you. So what about the masks? At the beginning, they were not allowed to be used because they were for sanitary people. After 1 week, it was compulsory to be used. But the science was saying that mask is not necessary. So be careful. I want to be, how can I say, to myself and to you, colleague, that keep in mind that it's very important to be honest in what we say. So don't miss that, sorry.
Yes, thank you for the question. I think it's very important also to focus on our wellness and that sort of stuff. First of all, I would like to share with you the idea that people tend to show off most their success without also being precise to show off also the weaknesses and the bad things that come across their own research. But on the other hand, when we focus on ourselves, we see both. So that's why you may perceive that your work may have been inadequate or different. And also there is a trend which may be more or less common among each researcher, which is defined busy bragging. So being proud of being very, very busy, highly full of stuff, and it may turn out being a toxic situation. For that reason, I think in my case, I'm trying to focus on daily work to fix realistic and short time decisions and milestones to focus, work, and see a trend of growth. Of course, sometimes there's no time or the possibility to take your time. But sometimes if you rest, if you take a breath and think finer, you think more precise, high workloads will be replaced probably by artificial intelligence soon. So in this context, maybe it's better that researchers do fine thinking, something that machines cannot ever do.
If I can tell a comment on what you say, one of my masters used to say, Ricardo, what is the gold medal? So how many ideas can you have in your life? He says Einstein may be at 3 or 4. How many ideas do you want to have? If you have one, you are lucky.
Only a quick question about, as most of you...
No questions. The
No.
questions will be answered by the next.
Oh, okay. So, I will not...
So I would like to report my experience when I was a postdoc about feeling not perfectly fitted for that work. And the boss used to come to me every week, every Friday, and say, remember, while you sleep, competition is working. That was helping very much.
We can stop here for this session, and then there will be another session now, and Gerardo can do the question after. We do the coffee break now, but please come back soon.
Yeah, sorry, I suggest a coffee break now, and the next round table group discussion will be called later. So I would like to ask you to take your time now to talk with first and second year students that are displaying the poster. Now we have the poster presentation with the coffee break in the same area. So that's the time to talk with the first and second year PhD student about their research. Thank you. And we start at 11:30. Thank you. Thank you. We go on with the next round table of the area agrosystem and bioeconomy. And the moderator is Dr. Valerio Mazzoni. And I would like to call the students to the stage. Paola Bettinelli, Anna Casari, Sofia Farina, Federico Grignafini, Daniele Prodorutti, Concetta D'Amato. So the students, we start with a very, very short talk about their topic. They give a very quick self-presentation, and then we start with the discussion. Thank you. Would like to start? Give me your name.
Here we are. So hello, everybody. I am Paola Bettinelli. I was working on grapevine during these 3 years, and I was facing the problem of emergent disease due to the reduction of pesticides. And what I was studying, it is black rot, that is an emergent disease, a fungal disease. And I was studying a segregating population to find new RLSI. And in the end, we came up with a really interesting conclusion that we have 2 loci, one that is associated with resistant to leaves and one to resistant to bunches.
Good morning, everyone. I'm Sofia Farina, and I'm part of the atmospheric physics group at the University of Trento. My PhD project is focused on slope winds, which are part of the thermally driven circulation developing in complex terrain. So these winds flow upslope during the day and downslope during the night, and are triggered and sustained by the temperature gradients that develops on the slopes. And we focus on the characterization of the turbulent structure of both the diurnal and the nocturnal components. And in particular, the diurnal components has never been studied before, so it was a very huge success. And we also focus on the applications of the studies of these winds in terms of the dispersion processes in which they interact, and in particular dispersion processes that are related to agricultural practices, and so dispersion of chemicals or passive tracers on pollens, and in general, whatever tracer.
Good morning, everyone. I'm Daniele Prodorutti. Today I'm here in the role of student, but I'm have been working here at the Fondazione Max since 2004, so a long time. And okay, I'm happy and glad to have the opportunity to carry out a PhD here. The topic of my PhD was the epidemiology of apple scab, which is the main pathogen of apple. Not only Venturi inequalis, but there is also a new species of apple scab reported a few years ago, even here in Trentino, which is called Venturi asperata. The main topic is to find sustainable methods to reduce the incidence of the disease, and we developed a method with the sprinkler irrigation in order to release the spores of Venturi inequalis during the primary season of infection. In order then, when there is the following rain, we found a low number of spores released in the irrigated plots, and even in the field we tested this system, and we obtained an efficacy around 50% in reducing the disease in the untreated plots, and also in the treated plots we obtained a high incidence of reduction.
Good morning, I am Concetta D'Amato, a PhD candidate, and I am part of the hydrology group of the University of Trento. Well, in my PhD, we studied the physical interaction between infiltration and evapotranspiration, so the physical interaction in soil, plant, and atmosphere. And we built a numerical model to simulate infiltration and evapotranspiration, considering some of the physical phenomena that occur in this system. considering, for example, the water stress, and so how water stress the evapotranspiration rate, and also the environmental factor as the different temperature of the air, the different vapor pressure deficit. We built also some a model of the solid transport in order to understand which type of water plant use from the different depth of the soil, using the isotope for the analysis. And nothing, this is the model that we built. So thank you.
Hello everybody, I'm Federico Grignappini. I'm a third year PhD student here. The aim of my project is the identification of the genetic regions on pear involved in the control of fruit quality and post-harvest related pairs, post-harvest related traits, in particular superficial scale. Basically, my PhD is split into two parts. The one in Spain was done in Spain, and the second one here. On Spain, it was the part related more with the phenotypic data, and here I will be able, hopefully, to do the genotypic part.
So thank you, everybody. So I'm Valerio Mazzoni, I'm an entomologist. So, I take the chance to start a discussion from the previous roundtable round, and what emerged was that when you're a PhD student, you have a lot of competition. There is a rush to publish, to have achievements, and however, yeah, this is true, but I think that an important point for PhD students is also to find what characterizes their work and what is the point of originality that makes you different from the others. And so I want to ask you to talk about this, how your work was different and is also a step forward from the previous research. I don't know who wants to start.
I really thank you for this question, because in my case, I had to face some problems collecting the phenotypic data when analyzing the population. And what I had to do was to go to the field to analyze it. And that changed all the story of my PhD, because I decided also to analyze the bunches that, of course, I was not able to study in the greenhouse. And it creates all the story of my PhD, because it is really rare in grapevine to study bunches, because it takes a long time to have bunches, like 4 years on a plant. And for resistance, even more, because when you have bunches, you study agronomic traits, but not resistance, because you don't want to infect your plant in the field. But since we have the disease here, it was possible to do it, and we had the population in an untreated field that is untreated since a long time. So that really changed the story of the PhD, and changed also the idea about the resistant loci in grapevine, because we were all thinking that what you find on leaves is the same on bunches, but it's not.
So in my case, when I started the PhD, we wanted to focus on the daytime component of the slope winds, and the original plan was to perform an experimental campaign and to collect data and then analyze data, and then use these experimental profiles and values to go further in the numerical modeling, the parameterization, and the analytical description of these winds. But due to the COVID pandemic, and also due to a lack of funds, we were not able to perform these campaigns, because these kind of measurements, including the turbulence component of the flows, are very, very expensive. So we were starting without data to try to understand this kind of flows. And so what we did was to try to take advantage of all the previous campaigns that have been done in the fields. So we started several collaborations, and we are now currently working with data sets collected by 5 different universities around the world. And so I wrote to those researchers and I told them my problem and what I wanted to study. And they gave me all the data that they had collected during the years. And I'm working also with pretty old data sets collected in maybe 20, 25 years ago, which is, I mean, the technology used then to measure turbulence structure was pretty different, but we can still we are still exploiting this data to their maximum to try to get the information that we need. And so in the end, we ended up using a data set that were considered like already exploited at their maximum, and we are still getting new informations and new results out of them. So I think this is a very huge success, and I think also in the community of boundary layer meteorology, this was really appreciated.
Okay, regarding apple scab, okay, it's one of the most studied fungal pathogens worldwide. So there are a lot of information regarding what happened during rain, but there are very few information on what happened when I carry out some sprinkler irrigation. And so the effect of this irrigation on the release of spores, because apple scab overwinters on the ground, on infected leaves, and so irrigating before the forecasting rain, there is an effect on the release of spores. And also there are very few information on what happened on the spores at different heights, and we also tested this. So it's something quite new. And as well as regarding the Venturi asperata, there are just a few, 2 or 3 papers on epidemiology also on this fungus. So everything it's new to understand the conditions for infections that are different compared to quite different compared to Venturi asperata, Venturi inequalis, and also the release of spores occurs later compared to Venturi inequalis.
Okay, well, I was born as an environmental engineer, but during my PhD, I used my knowledge on the physical part of the soil-plant interaction. But then I learned the informatic part and the informatic world. And so what we did is focus and create a model that is not simply a model, but is a platform. Because the idea is give to the user the possibility to decide which, for example, evapotranspiration model want to use. And in the decision of which type of evapotranspiration model is also the type of the detailed analysis that you want to do. Because if you want to do a very detailed analysis, you can use a energy balance model. But if you want to have a very simple analysis, you can have another type of model based on an empirical equation. And nowadays, there is no one model that could have the possibility to change in a very simple way different type of evapotranspiration model, infiltration model, if you want to use the route or if you don't want to use the route. So the idea is create a platform in which the users can decide which model they want. And everyone can add, because it's completely open source, so everyone can add other type of model, of course related with the soil plant, in order to create a system that everyone can use. So this is the idea, and of course in the behind there is also all the physical part, but I think that the main and the different characteristic is that.
Well, I am working on PEAR, as you know, and it looks like in the world no one or only a few guys are working in this topic. I mean, PEAR is related with Apple, but even if you search in the literature, you can find only old papers. So, well, I think the working on this topic, I hope, I strongly believe, can add some more information to breeders in the future to find again this, well, not find, to give again to this crop what it was in the past. And the knowledge, well, I am working on superficial scald. It is not so understand what is the pathway and all the biochemical things underneath of these diseases, this disease. So I think, well, a great opportunity, a good opportunity to work on it and to share one day this information to other people.
Okay, but I would be more curious to know if now, at the end of your PhD path, can you do something that makes you really peculiar? Something that you are one of the few people in the world can do, or not? I was feeling like that when I finished my PhD.
Oof. So, if I have to think about myself, well, of course, I am a better person, let's say. So I can give to other people, I don't know, advices or other things. At this point, for the science, for the word science, I don't think I am able, but I am not at the end of my PhD, so I am at the beginning, the third year. So in this year, I can not make the difference. No, I'm not using Bolt, but I can do something well for the other people.
Well, it's the same. Well, I think that I don't have the knowledge to understand if I will be a different person, or maybe not the knowledge, but I don't know if I can do something of different. Maybe the other can understand if I can do something of different, or maybe judge about if I can do something better or not. What I can say is that as Sicilian, now I can prepare a polenta, for example. So now this is what Trento, or I can go to the mountain and burn my face, but it's another side. Well, from the physical point of view, I think that I am a very curious person, and in everything what I did is that. So I hope that this curiosity can help me in my research and in my future in general.
Okay, I think that with my research, we developed something new, and we have also the good results. Also, when I presented the results in some meetings. So there is something new on Apple's CAB, and the results of this project can be easily applied in the field, and this is very important for me, and I think it's a good result. And okay, that's it.
Change slightly the question to the next student, and it's so you feel you reached the achievements and the goals that you had at the beginning of the PhD.
So in my case, I would say yes, because the real aim of the work was to produce a molecular toolkit for the breeder to identify this resistance loci. And I was really happy to have a huge feedback from breeders when I was last years in Landau in a conference. So we got in contact with them, we developed the molecular tool, and they are now using it, and they will let us know if it is working well as we hope. And so in my case, I wanted to have a really applicative outcomes of my PhD, and I reached it, so I'm really happy about it.
I think that also the objectives of the research project changed a bit during the PhD. So maybe we're not getting to the objective that we defined at the very beginning, but we are getting to the objective that we defined during the course of the PhD. And we still have some months to work, and so I'm confident that we will reach them. And also related to the previous question, I think that this this idea of being like the one of the few people in the world expert in that field is also like it's a component of how the science is working now in the world, and so this hyper specialization that we are facing. And so it's, I think that at the end of the PhD we are like the people that are the most expert on that topic, because we worked only on that topic for 3 or 4 years. So I think that right now I am probably one of the few people that are very, very expert on anabatic winds, because I worked only on anabatic winds for 4 years. And so it would be a problem if I was not. And so, yeah, this is a bit like part of the way science is working.
Yeah, that's exactly what I meant. So, 10 minutes. It's enough. Is there any question from the audience?
How do you ensure that you label for example, gravitational water or the adhesive water in the soil, and not label the other types of water present in soil because of the physical forces that are happening between those different water types, interactions with soil, etc.? How do you know that you label, for example, only one category and not the other ones?
So the point is that this is the simulation, so all the experimental part with a limiter. The limiter is a very constant system. So in that case, we can write that this is the basic. We are making sure of the that they take. So the system is controlled. I don't know, and I don't know because I am in another field and not in the practical field. Yes, sure, yeah, you're right. For sure, you're right. Like,
Good. Other questions or comments, thoughts? Okay, so then I go further, and my next question is my point of discussion is if you feel now, this is not a question for Daniele maybe, but for the others, if you feel like to be ready, to be independent, to be a scientist, a postdoc or a researcher without a supervision, do you feel like?
Well, yes, comparing to the when I started, for sure, yes. And I hope to demonstrate that in the thesis discussion one day. Well, comparing with when I started, I think yes. Also because I think that the PhD is not only a research period, but it's a very strong life period in general. And yes, during this PhD, everything happened, COVID, disease, everything. So I think that it's a very strong period that you develop in a very different way. And so, yeah, I think yes, but I don't know.
Yes, I'm ready. Okay. No, yeah, sure. No, anyway, it was for me a good, very good opportunity to open my mind. I was we were very interesting also the courses that I followed during these years, and I learned a lot of new things that I can now apply in my job. So very useful.
So also for me, the answer is yes. And also because during the PhD, we do not only learn about like coding or doing experiments or measuring data and plotting them, but also how to interact in the scientific community. So how to present in conferences, how to send an abstract, how to start a collaboration with another institution, or I don't know, there are so many collateral things that we learn. And I think that after 3 years now, I feel a bit more confident also in like speaking in front of an audience, or yeah, starting collaboration, or also maybe writing a grant, or so, yeah.
It's also my case. I grown a lot, and what I want to say is that the next step that I'm facing now, it is to work on different projects. I mean, now I don't have just my PhD, but I have different topic that I have to follow, and this is the new thing that I will learn applying what I learned now.
Great, seems that it was really important for all of you. And yeah, importance of networking, that's very important for you. I have a couple of minutes, and
so listening from your answer, and also the discussion that we had before, we came to my mind one aspect that I wanted to listen to your opinion. So the contrasting necessity from one hand to focus, because you have a project and you have to focus, but also the realization, especially at the end of your experience, to the fact that the problems are more complex than can be solved by just focusing on one aspect. So how do you deal with this, or did you start thinking about this? What do you think about this problem now?
chi risponde?
Yeah, in this contrast, I think the key is that we know that we are very, very specialized in one topic. And so if we collaborate with other people that are specialized in the other topic, that are part of the main problem that we want to solve, we can use all the different knowledges and reach the goal together. Because yeah, it's impossible to both focus very deeply on a specific problem and then keep an open eye on like the general picture. I mean, we try to do that, but I think that collaboration is key to maintain this greater view.
Well, in my case, the answer is also informatics because, the answer that we give is we know that physics, and in particular physics of the plants, is continuing in developing. And also we are implementing new equations that are different from the equation that I'm still implemented. And so the point is create a way, and from our case, the informatic point, to change in a very simple way the model that we want. So we study and we continue to study the physical part, and in parallel we create the system to compare every day and every time the stuff.
Last one, and in my case, I'm really a curious person. So what I wanted to do during these 3 years was to learn techniques that would make me able to face different topics. So in these 3 years, I learned many techniques that can be applied in plant science, but not only. So this is what I think.
Okay, the session is over. Thank you very much. Thank you to the students. Thank you to you.
Thank you. We go on with the very fast with the next speech. I would like Professor Rigon to introduce the next speaker, please.
It's difficult to me to introduce Cristina Rulli because first she is a friend and then comes the scientist. But she is a fast-seeing scientist. She covered all the topics of surface waters in the past, which also I covered, and she probably did better than me. So that is the introduction. And she is professor at the Politecnico of Milan. So when you say that, you say, I am professor in Berkeley, if you are in the United States. So Cristina introduced herself. The topics in which she worked recently are very, very exciting for the complexity of the argument, which is the soil fertility, the interaction between water and agriculture, the grazing, and many things that also I don't know because I am curious to see now. So that's what can I say. So looks at her presentation, I think that you will learn also many things. And I also, maybe casually, but the topic she is covering is very much in line with what we already have seen. So there is a possible, let's say, hanging between hydrologist and genetist.
Thank you very much. I hope you don't have too many expectations after after the words given by Riccardo. So at first, thank you very much for being here. And it's a pleasure for me, and especially it's a pleasure to meet you and my colleague and friends from University of Trento. I learned a lot today from your presentations and from the presentation given before about genetic. And I learned you have very diverse backgrounds, so I hope my presentation will be interesting for you. So the title of I don't know if it is okay. Well, the title of my presentation is Connected the Dots Between the Water, Food and Energy Nexus and also Human Health. So one of the main challenges we are facing nowadays is to feed a growing population that is expected to be around 10 billion people in 2050. These people is demanding for additional 60% of food, 20% of energy, food and energy, sorry, 60%, 20% of water, and 30% of agricultural land. So exacerbating the competition for the natural resources between the different sectors using natural resources, especially food sector and energy sector. Additional challenges are related to the eradication of malnutrition. That is represented by undernutrition, but also by micronutrient deficiencies, overnutrition, that all of them in turn affect people with food-related diseases. Moreover, what we are looking to is that some countries
that are becoming more affluent are following the so-called Bennett's law. People from economy are very familiar with this law. And so these people, these countries following this Bennett law are over-increasing meat consumption at the expenses of a balanced and healthy diet at first, but also at the expenses of a sustainable food production in terms, of course, of use of natural resources, especially water and land. We are, in fact, observing, as you can see from the plot on the right, a sharp increase in the last decades of the incidence of the emerging infectious diseases, especially of the zoonotic diseases, meaning that the infectious diseases coming from animals. And most of them are related to an unsustainable land use for agriculture, where agriculture has to be intended both in terms of crops but also in terms of livestock. If we look to the history of food security, we see that the debate around food security shows us that the question of feeding growing population is not a new question. In fact, Thomas Malthus at the end of the 18th century analyzed the availability of natural resources for feeding the growing population and the availability of natural resources for producing food. So predicted in the long run the insufficiency of food to feed everyone, the so-called Malthusian catastrophe. Malthus' predictions did not come through. We were and we are lucky. And Esther Boserup ascribed that, so the escape of the Malthusian catastrophe, to the technological innovations that allowed the humanity to increase food production more than expected by Malthus. The mechanization of agriculture from one hand and other innovations bring by the Green Revolution boost crop production at a point that global food security was not a problem of only food availability, but also of food access. As Amartya Sen, that was a Nobel recipient, point out. But now the question is to what extent is current food production sustainable? And so the question, how many people can the planet feed, is still relevant. If we look to the strategies for increasing food production, the main two strategies are called agriculture intensification and agriculture extensification. Agriculture intensification means to increase crop production while keep constant the cultivated land. So intensification implies the boost of crop production through the use of the so-called agricultural inputs, especially irrigation and fertilizers. So increasing the agricultural yield and closing the yield gap between the actual yield and the optimal yield in the underperforming croplands. The agriculture extensification, by contrary, means the expansion of the cultivated area, so not increasing the use of agricultural inputs. Both the strategies have effects on the environment and also on the society. And about agriculture intensification, we can have the transition from from small farmer agriculture to commercial agriculture, the loss of livelihoods, but also the water resources depletion and the land degradation. About agriculture intensification, we have problems connected to land use change, deforestation, and biodiversity losses. So usually ecologists advocate for intensification rather than extensification. The main reason why is because intensification avoids habitat destruction and land use change. But we are hydrologists. I am hydrologist by background. And so the first question to be addressed is what about the amount of water needed to boost crop production through intensification? So what we have done was to model crop production at global scale in a spatially distributed way, so using an agrohydrological model, with the aim, with the goal to evaluate the amount of water needed and also to evaluate the amount of additional crop, so additional food that could be produced by intensification. Here you see that we analyzed, of course, different scenarios in our analysis, in our paper, but what it was important, the main result is that about additional 4 billion people at global scale could be fed by irrigate crops when needed. But another problem arose once we did this analysis. And so, do we have the natural resources to sustainably feed the humanity? And so it means, do we have water to irrigate in a sustainable way the crops needing of water to boost their productivity? And the answer was that yes, our calculation gave 4 billion people, but we have places in the world that you can see here that are hotspots of unsustainable water use right now. So basing on the irrigation they are doing right now. And so it is very important to pay attention to the sustainability of the irrigation. And so what we did was to repeat again the analysis we carried out. So this time to pay attention to the sustainable use of irrigation water. At first, what we did was to analyze very in deep the croplands in terms of water scarcity. So we analyzed at first the lands needing of water, but then we analyzed if the water that could be used for irrigation is water that is in a place affected by the so-called economic water scarcity or physical water scarcity. Economical water scarcity means that there is water, but there is the lack of the hydraulic infrastructures for using the available water. So in those croplands, it is possible to make sustainable irrigation. But there are other croplands that are in water scarcity and are affected by physical water scarcity, meaning that in those croplands, the additional use of water is unsustainable for the environment. For example, those places are places where irrigation is done using fossil groundwater, for example, or exceeding or not preserving the so-called environmental flows. So if we take in account of the sustainable and unsustainable use of irrigation water, what we discovered was that we move from the additional 4 billion people that could be fed doing intensification, we decrease to 2.8 billion people. That decrease to 1.8 if we decide to eliminate irrigation in the places currently doing unsustainable irrigation. If we want to look to the number of the sustainable irrigation expansion, we can see here the hectares that can be irrigated at global scale in order to increase food production for in a sustainable way for the 1.8 billion people we talked about before. And we see that, of course, the number of hectares of land that could be irrigated increase if we decide to increase to irrigate not at the potential but to a 20% deficit, and increase more if we consider to irrigate at the 50% of deficit irrigation. That are strategies of irrigation, of course. And so we see also the associated amount of water needed for the intensification of agriculture. It's important also to look to see where it is possible to do this intensification. We have, of course, the results at global scale, pixel by pixel, but it's very interesting to see the results reported at country scale or at group of country scales. And here you can see that the Eastern Europe and also the Sub-Saharan Africa are two regions where intensification is particularly could bring a lot of food, but we know very well that Eastern Europe in this period is not the best place for intensification, but it is not also not the best place for producing food, and it was one of the main breadbasket of the world. And so we have to keep in mind that this world can to this work can be associated also problems in terms of food security, not only for Russia and Ukraine, but also for the countries linked to this, to Ukraine and Russia through the import and export, to the trade. Here we have summarized in terms of calories and in terms of people that can be fed by the intensification. Of course, the climate change can exacerbate of course, in some part of the world, water scarcity, and so we could have effects also on the additional amount of food that can be produced. There are also other effects, both in terms of social effects and also environmental effects associated to the intensification. And here I reported, but I cannot go in detail, a phenomenon called large-scale end acquisitions. This phenomenon, negatively termed land grabbing, started in around 2007-2008 during the financial crisis that then translate in food crisis. What happened was that corporations, international corporations, but also governments started to buy large tracts of land outside the country of origin. And so here I reported the network of the acquisition, where you see reported with the circle the land, the places where land were acquired, and with the red triangle the place where the investor came from. This the reason why the countries, the foreign countries started to buy these large tracts of land was because during this financial and then food crisis, they realized that food trade could be not the solution for a real food security for domestic food security or energy security. So they realized that the best thing was to have direct access to the natural resources for producing, in general, crops. And so there is the reason why they're starting to buy large tracts of lands, usually where water was and is available. And the situation now is that the total amount of land acquired is more or less 4 or 5 times the area of Portugal. So you understand that the problem was and is huge. And it's an environmental problem because here the investors started to do a very aggressive agriculture, so very intensive agriculture, don't pay attention to the environment. And from social point of view, it's a problem because associated to the acquisition of land, there is the dispossession from the previous users of those lands. The reason why is because usually these acquisitions happen in regions where there is not the title of land, so it's very easy, the dispossession. And about the water associated to this phenomenon, up now is 10 11 times the volume of the Garda Lake, just to give you an idea, not in number, but in something that could be understood very easily. Okay, 10 minutes, okay. Of course, additional challenges to the use of natural resources is given by the energy sector. And the reason why is because more or less any type of energy needs or water to be exploited. In here I reported just the biofuel, that it is really very debated during these days, this week. And here I reported the scenarios given by the International Energy Agency for the period 2016-2030. Imagine that those are the sustainable development scenario reported by the International Energy Agency. And in any case, even if this scenario is a sustainable development scenario, so you see the increase of water, this scenario report. And I show you this, but we did in these years a lot of analysis regarding the use of natural resources by the energy sectors. I reported this one because not having time to show everything, I think that bioenergy, biofuels are an emblematic example of the competition that can arise between food sector and energy sector. The reason why is because the feedstock is the same. Crops used for biofuels and for energy, for food and feed are the same of the crops used for biofuel. And in fact, there is what it is called the food, energy, and water nexus among the sectors, meaning that a nexus exists among food, water, and energy, so that those three resources are interlinked, meaning that energy and food are based on the same pool of natural resources that are water and land. And this is why the competition can arise if a unsustainable use of natural resources by one of the two sectors happen. Agriculture is one of the main driver of land use change, and here in this map we see the land use change associated to agriculture in the last, I think, 30 years. Why it's the land use change is very important? Because land use change is also associated with infectious disease emergence. And an unsustainable land use change in response to an increased demand of food can increase the proximity of humans to the wild habitat. It's what happened with the agricultural expansion, with an agricultural expansion implying deforestation and in particular forest fragmentation. In this case, we notice the encroachment of humans in the wild habitat. But if it happens mainly with agricultural extensification, it can happen also with agriculture intensification, consisting in intensive farming and intensive livestock husbandry. In this case, we have that the encroachment of humans in the wild animal is combined with the presence of the so-called generalist virus host species that are species that are able to survive in disturbed habitat. So humans disturbed habitat, but species hosting virus are able to live there. We investigated where the unsustainable land use change can enhance the risk of coronavirus spillover. And we carried out an hotspot, cold spot analysis in the rhino-fide bats suitable areas. Rhino-fide bats are considered the putative hosts of the coronavirus. And so we consider spatially distributed indicators of land use, such as land cover, the density of different type of livestock. Then we consider croplands, the distribution of croplands, the population density, and also human settlements. We also calculated, starting from land cover, from forest cover, forest fragmentation, and then we did a sort of multivariate hotspot, cold spot analysis, searching hotspot areas representing the encroachment of humans and animals into the fragmented areas. So from a mathematical point of view, what we did, we searched areas being hotspot of fragmentation, but also of density of population and also of livestock. This is the results. We studied an area of around 28 million of square kilometers, and we use a very high resoluted data regarding the indicators. And so doing this analysis, the hotspot, cold spot analysis, here the results we obtain. It is interesting to see that China is a very, very big hotspot, but it's hotspot also, this part of India. It's very, very interesting also this, because Nipah virus usually happen in this area. Then we have something in Pianura Padana, and also in this part of France, that it is Normandy, I think. And the last, I'm finishing, the last exercise we carried out is to look the places where we are not in hotspot condition, but where unsustainable trajectories of land use change can bring the place from neutral to hotspot. And here some example of those places. So going back to Malthus, we still have to prove Malthus wrong. because the challenges of food supply are a major and continuing challenge facing humanity. Because even though global agriculture production can feed the planet, it does do not sustainably. In fact, what we can see from the food balance sheet provided by FAO is that more or less we could have enough food for feeding everyone. But the problem is that a lot of food is not produced in a sustainable way. And so if we look to this from an environmental point of view, it means that we have to move from an unsustainable food production to a sustainable food production, taking account not only the nexus between water, food, and energy, but also the nexus with health. Here, some sustainable strategies for increasing food security. In red are the strategies we are working on with some of our students together with Paolo and of course our students. Paolo is not a student. And so here we have an example, so we can make sustainable intensification, but also we have to reduce the demand of food, because reducing the demand of food by changing the diet, we reduce the pressure on natural resources, and also reducing waste in terms of food, we reduce waste in terms of natural resources and also in terms of nutrients, in terms of the inputs we are now using. So here some example of the analysis we are doing now. And so just to conclude, food and energy production have an impact both on environmental sustainability but also on human health. So we need to combine the food, energy, water nexus with the One Health paradigm. So thank you. And so if you have questions, I'm here.
Are there any questions from the audience? So I will start. Okay. I have many questions, too many questions to. One is very simple. Why hydrologists now urge to work on those different? We was born as engineers, as Concetta said before, and now where we are going?
But we are engineers. We are engineers also doing this type of research, I think, because we are using our brain. The meaning of the word engineering comes from ingegno in Italian. And so it's very important that scientists in general, but also engineers, of course, analyze this type of problems, because we need to design strategies to save natural resources, to preserve our environment for the future generation. So the definition of sustainable development is that we have to preserve natural resources for the next generation. Then to do that, at first we have to think in a nexus way, because the resources are interconnected, are interlinked. So I cannot think to use natural resources for one sector, because associated to the use of natural resources in one sector, it means that the other sector cannot use the same resources because the pool of resources is the same. And so engineers are really very, very helpful in finding solution. And beside this, what I want to say is that to put together environment and human and animals' health is very important because they go together.
Maria Cristina, I enjoyed very much your talk. It was very, I learned a lot and it was very intriguing. A couple of questions I have is about your scenario is based on the plants and especially the plants. So I don't want to talk about animals, but the plants that are not selected in a different environment. Because one of the slides was about plants with a better water efficiency, water use.
Yeah.
And I would like to comment, not necessarily transgenics. Transgenics that don't kill people.
No, we didn't consider transgenic
just to be
plants.
sure that transgenic don't
yes,
kill people.
in
Europe that we cannot use it, but maybe in
this
some other
one,
part of the world.
plant the right crop in the right place. What we did here was to consider the crop planted on a cell that is 10 kilometers per 10 kilometers. In each cell we have different crops. So what we did was to try to see if planting the crop in this grid in the right place, so it means in the place where there is water, precipitation water in the right time for the growth of plant. If it was, we checked if it would be possible. Checking that, so planting the right plant in the right place translate in water saving. The reason why is because if you plant a certain type of crop in the place where this crop can grow using precipitation water only, it means that this crop doesn't need of irrigation water. So of course we are hydrologists, so all of our calculation have as goal minimizing water use. And about the suitability of land, we hypothesized in this exercise that in the grid, land is suitable more or less in each place. Now we are doing more or less the same, but adding also as goal not only minimizing water, but also providing the so-called the Lancet diet, that is a special type of diet considered also healthy and well balanced. So what we are doing now is to look where to grow crops, satisfying the minimization of water, but also pay attention to the suitability of the soil. In this exercise, we are looking also if the soil is highly suitable for the plant we want to, for the crop we want to plant.
One comment. I was also thinking about the climate change, which was not considered by you. Let me say, and then the other question is, the way agriculture is using water is also not efficient, because, for instance, in the Po Valley, for instance, most of the water is used by dispersione. I don't know.
Farrow, yeah.
That's depending on the special crops they are using, and maybe the engineering They
Yeah.
should develop a new system of like the drop irrigations for some of these crops.
Well, this is another story because then we have to talk of the irrigation water system and the efficiency of the irrigation water system. And so improving the efficiency, we theoretically we could save water. I'm saying theoretically because the extra use of water we withdraw with respect to the water needed by plant is water that return to the environment, and sometimes it could be really beneficial to have water into the environment rather than, so there are calculations that can be done. In any case, yes, sometimes the irrigation plants are not efficient, but in this direction we can do something, but we have to do something also in other directions that are the direction I showed before.
Thank you, Christina, for the answer. Thank you, Mario, for the questions. We don't have very much time, but the discussion can continue later. Now I introduce Paolo Dodorico. Yes, Christina is a friend, but Paolo is a brother because we share it. So now she is jealous because we share the two of our master, Ignacio Rodriguez Iturbe and Andrea Rinaldo. And what can I say of now I have a small time because he's uploading the what can I say about Paolo? Paolo is also an hydrologist. He started to work on the hydrological response. Actually, we did some work together many years ago on the hydrological response, meaning how water after the rain, what does water after the rainfall? The question that Mario did before, as Christina also told, actually has complex answers because not always, for instance, the irrigation that save water for the environment, for instance. And there are feedbacks that, so Paolo is ready now. Paolo is professor at Berkeley. He was also director of the environmental science departments. And by himself, he's one of more productive hydrologists in the world, meaning that his publication is literally huge. So you think that one that is doing an industrial number of publication is producing low-level science, then the issue is here that his science is also very high. So I am happy to have him here to talk. So now, Paola, are you ready? At the present, he's doing his sabbatical at the FAO, but now you tell yourself. Go ahead.
No, thank you for the introduction. I'm going to sit down because I have to talk for the microphone. And I was trying to change the light because I felt there was some glare, but I hope you can see. As you can see, Christina and I work together a lot, so there will be some overlap or some continuation. The focus today is again on water in agriculture, and most of the water we use as a society is for agriculture, for the production of food. And there is also the second sector that uses a lot of water is the energy sector, but as you can see, is much less than the water used in irrigation. And so this still locally can be play an important role. And Christina already showed that there is an increasing demand for food, and this means that there is an increasing demand also for water, because again, most of the production of food really requires water. And this is again, the increasing demand is because of population growth, which has been allowed partly by many factors, but including the ability to produce more food through the innovation of the industrial, the green revolution, global trade, and so on. And through some of these changes, some of the limiting factors have been removed. The typical example is the industrial production of nitrogen fertilizers, but water remains a limiting factor. And then in addition to this, there is this change in diet, which is a diet richer in meat that requires more resources, including more water for their production. So let's look at where this water is coming from and how the water system works by looking at water over land. So water leaves continental land masses in two ways, either as water vapor, and most of that water vapor is mediated by vegetation, or in the liquid phase as surface and groundwater runoff. A few decades ago, this jargon has been introduced. It's pretty common mostly in the water sustainability debate. Green water flows are referred to as evapotranspiration, and blue water flows as surface and groundwater runoff. If we look at agriculture, we have most of the world is agriculture is rain-fed. And this means that it uses only green water flows. Instead, part of the land, about 20%, is irrigated, so it uses both water coming as a precipitation and water that is comes from irrigation. Irrigation takes part of these blue water flows and converts it into green water flows. And so the global evapotranspiration is this big number. Part of it is in natural ecosystem, partly in agroecosystems. Natural ecosystems are not irrigated, so they just use green water flows. And instead, the agroecosystems use green water flows, but partly also blue water. And so this is a tiny fraction, but it's still very important. Many rivers in the world are running dry. I always talk about the Colorado, the Rio Grande. They don't reach the ocean because they are strongly depleted. They are basins of internal drainage. The Aral Sea, Lake Urmia, Lake Chad that are shrinking because water is withdrawn from the rivers that flow into these lakes. Groundwater is also strongly depleted. And so Christina already showed regions of the world where there is, this is based on the water balance, where the consumption of water for irrigation exceeds the local runoff minus environmental flows, which means that if there is a deficit, it means that the water is either taken from groundwater stocks, so from the next generation, or from the environment at the expenses of environmental flows. And we also live in a world that is strongly interconnected. This is the current situation, this panel, and then we have different scenarios of population and growth and climate change. And we can see that the ability to produce food is the ability to produce food, which is often limited by water resources, and again, Christina showed something about that, is limited in some areas of the world. Some areas of the world are affected by a chronic water scarcity, and as a result, they are not self-sufficient. And the response to this has been the development of global trade, which has been increasing since the 70s to up to now. And from the mid-80s to 10 years ago, it more than doubled. And we see that these are the patterns of global food trade. We see that over the years there has been a more a stronger integration of South America in the global trade, and in particular the emergence of this strong trade and can be the famous soybeans, which are used as feed for livestock in China. And many other parts of the world the world has become much more interconnected and with stronger trade dependencies, to the point that if we now calculate the minimum distance from which different commodities, agricultural commodities reach different parts of the world, we see that many parts of the world, the distance is of the orders of thousands of kilometers. So as much as we can be supportive of the notion of local food and local food movements, the way the food system is structured now is strongly globalized with these strong interconnections and interdependencies. Of course, a few things can be changed because some part of the food system has been developed without using local resources, because the local systems of productions have been displaced by cheaper commodities coming from other parts of the world where they were strongly subsidized. But at the same time, and the population has also grown sometimes in areas of the world relying on food produced somewhere else, and also at the same time relying on water resources existing on the other side of the world. So associated with any food commodity, we can think that there is also a certain amount of water that is not physically present in the commodity, but went into the production of that food product. And so that is usually called virtual water. And the idea is that associated with the food trade, there is a virtual water trade, which allows us to show how parts of the world depend on water resources existing on the other side of the world. So the food system and also the water system is strongly globalized. And to date, we have that about 25% of the food and the water used in the production of food is accessed through international trade on average. These are the premises of the food system that is strongly globalized. It is locally affected by scarcity of water. Water is used mostly for the production of food. And so we are convinced now that water security means food security. When we talk about the food crisis, many times there is behind a water crisis, but for sure, if there is a water crisis, most of the times is because there is a lack of water to feed the people before having a lack of water to quench our thirst. In the sense that we need much more water to feed than to drink, and to allow to quench the thirst. So human rights are strongly coupled to water security. Firstly, through food security. So what are the challenges? The challenges is there is always a debate of whether water is a good or a human right. What are the mechanisms of access to and appropriation of water resources, and how we can interrelate issues of sustainability, justice, and inequality. So I want to stress, first of all, the relationship between water and water needs, and the water needs there is often in this debate a very strongly anthropocentric perspective. We hear it all the time, right? Our environment, our climate, our water, as if humankind had everything, all natural resources were there at the disposal of humankind to meet and instrumental to our needs. Instead, there are also needs of nature of the environments to function properly. If we want to preserve aquatic habitat, rivers need to maintain a certain amount of water, for example. And so in recent years, there has been a strong debate bringing into this discussion the notion of the rights of nature, which should be sort of the counterpart of human rights. So nature as having some sort of personhood that can claim its own rights, and the rights of the environment, the rights of nature, recognized as something that is important from a more ecocentric and not anthropocentric perspective. Then in the terms of human needs, we can divide two things. First of all, the water we need to feed the world. And we saw that this is a short blanket. Christina's talk already showed that. And so the water needed to meet human rights. In addition to that, we need water for many other things, in smaller amounts, however, and to sustain businesses, to sustain different enterprises. And so next to that, there will be some sort of commercial water rights. And today, I want to bring up the question of whose water again. And again, if it's the first question is whether it's of the environment of humans, and then if we are in the context of humans, how do we adjudicate the ownership of water? So this is a whole field that is emerging in recent years. It's called the field of water tenure, in the same way as we have been talking about land tenure systems. Who has rights on water? Are these rights pertinent to the land? So do they come with the land or can be separate from the land? So, and then sometimes these rights are defined separately from the land. And what do these rights include? And so typically in the definition, and if there are lawyers in the room, probably they can explain this better, but the rights, in particular property rights, come with some sort of a bundle of rights, depending on how strong these rights are. Can be just a right to use a resource. There could be a right of not to use the resource, because many times if you don't use the water, you lose your water right. The right to transfer your right to someone else. Can you sell it or not sell it? So this means that you can create markets, and there are water markets around the world in which there are water entitlements, which will be like the title you have on your land, or on a piece of land. You could have it on a water right, and you can trade it, and this creates a market. And this is a pure modification of water resources, because often markets function with the purpose of creating, generating revenues. And then the right to exclude other people from having access to water, the right to manage the water resources that are given to you, or to have a voice in their management, or to appeal for the decision of a water authority about your water rights. So these are different ways to characterize whose water and a water tenure system. So when we have that rights, water rights are pertinent to the land, they, typically, this is typical of many parts of the world. And so it means that these rights are acquired through a land acquisition. If they are separate, we can have for the some water rights and define in different ways whose property are they. So it can be a property of the state, and then the state then assigns these rights to use to someone else. Can be a private property of an individual, and can be a common property of a community that then decides internally how to manage, how to assign the use of these rights, or can be open access, so everybody who can go and get the water without particular regulations. This means that there are different mechanisms of acquisition given a certain water tenure system, which can be through a concession, like I think it's here in Italy, a concession, a license or permit to use water, or if it's private property, it can be acquired, and so there is a market. And if it's a common property, there are some customary arrangements, and otherwise it's completely unregulated. So commodification of water at this point becomes, requires, first of all, the recognition, acknowledgement of some private property rights on water, and these private property rights need to be tradable. And so this is, you see that there are often some concerns about that. So we were at the UN conference in New York, and there was a conference on water. And in the same time, they were making announcements acknowledging that the water, or they were calling the water cycle, whatever it means, is a common good. And at the same time, they were claiming that the solution to the problem of water, going with the old rhetoric that water is wasted because people don't pay for water, we need to privatize and commodify water to solve the problem, without thinking that there are impacts on subsistence farmers, on livelihoods, or it's much more complicated than that for sure. But so there is always often this advocacy for a commodification of water, and at the same time recognizing that water is a human right. And so these are parts of the world, for example, in blue, where there are some water markets. And I like to present this quote here. Is in a book of political economy. It's a book I've been reading. It's that every society, Harvey says, sets boundaries on where commodification begins and ends. And then it also clarifies that there are some places that are really borderlines. And his examples, if you allow me, he was saying that in some countries, sexual favors can be commodified, and legally in other countries it's not possible. Water is also at that boundary. In a sense, in some countries it is commodified, in other countries it's not, as this map shows. So the thing effect is that by looking just at rights, sometimes we don't capture the complexity of claims that can exist on water. So even if a legal access to water can still violate some customary rights and some access rights that have been established by long-term habits and customs. And apparently, the FAO did a study, 50% of farmland area in the world has access to water through these customary arrangements. So just looking at formal law is not sufficient. What is interesting for us is now to connect these mechanisms and legal mechanisms of access to water to some more hydrological and biophysical mechanisms and see how we have access to water resources. So we just said that we have access to water, for sure to rainwater by buying land because it rains over that land, but also to the blue water through some mechanism through an acquisition of land, particularly if the water rights are pertinent to the land. Then other ways is to gain control on water resources, because now how can I use and exclude someone else from any access to that? It's through infrastructure. So if I build a dam on a river, or someone does, of course, can be the government, can be an agency, etc., I control the water, and then I control the access to this water. Similarly, can be a well at a smaller scale, can be a diversion waterworks or something else. Then we can affect and appropriate and exclude others from any access to water is through pollution. Because in a way, even just a small amount of pollutant is tolerable by the environment if it remains below a dilution ratio. But it means that someone else cannot withdraw water from that river beyond a certain level, otherwise the concentration of the pollutant would increase. So if someone pollutes, it means that there needs to be a water allocation in the stream, a fraction of the flow, available for the dilution of the pollutant. And so, without even going to the more catastrophic situation of dumping toxic waste, so nobody can use that water. Then there is another way of appropriating water by planting trees or changing crops, by using plants that are more water demanding. And finally, through more efficient irrigation systems, we were talking about the drip irrigation before, we can see more of that. We have studied over the years a phenomenon that we defined as water grabbing, which is an appropriation of water in conditions of unbalanced power relations, in which there is asymmetry of information, asymmetry of power, in this regard of the environment or previous uses, in conditions also scarcity when there isn't water for everyone. And some of these mechanisms of access to water can really lead to water grabs if these other three conditions are met. And so I won't talk more about land acquisitions. We did quite a few studies. In this paper, we gave some examples also. But most of the times, large-scale land acquisitions, Christina was referring to them before, tend to occur in areas with preferential access to either groundwater or to surface water, closer to streams, or sometimes even adjacent to the streams on the banks of rivers, or in areas with low green water scarcity, so in areas where already there is good rainfall. And let's look at the appropriation through dams. So this is, I'm going to show some example. This is the case of the Omo River, has been often discussed. It flows down to the Lake Turkana, the border with Kenya. And the construction of the Gibe 3 dam led to the emergence of plantations in the surroundings, but the downstream populations were left without water. And these are pastoralist groups who don't have any sort of recorded water right. And also fisher folks on the Lake Turkana are affected because of loss of fisheries in the river. We did a study on the construction of several dams in Africa since the year 2000. We looked at the emergence of irrigation areas. This is an example. This is before the construction of the dam. After the construction of the dam, there is the emergence of an irrigation scheme. And trying to answer the question, who benefits from the dam? Are smallholder farmers or large-scale agribusinesses? So because many times there is this other rhetoric that dams are used for economic development of rural communities. But what we found is that by looking at the size of the land parcels, there is an increase, not in all cases, but often the establishment of large-scale farming in these irrigated areas, suggesting that the benefit is for large-scale agribusinesses, often foreign agribusinesses, not for rural communities. Yeah. 10 minuti, ce la faccio tranquilla. So then afforestation is another example. By planting trees, we reduce runoff downstream. This is what we teach in forest hydrology all the time. So we did a study. These are based on areas defined suitable for tree plantation in around the world by the study of Bastian. And this is a study done by one of Christina's students. We looked at what is the impact of planting trees there. And this is the emergence of water scarcity, which means that there is a decrease in downstream runoff in competition with adjacent farms in the area. So all these grand ideas of solving the problems of the world by planting trees and for climate change mitigation, etc. Clearly, water is not being included in this type of analysis. And also the fact that someone else who used to have access to that water, these are customary rights, most likely, they lose access to that water. Whose water? Depends on the type of water tenure system. In South Africa, there are regulations that require a water permit to plant a forest or just a tree plantation. So this is, there are feedbacks on precipitation by planting certain crops or just afforesting a certain area. We can affect precipitation somewhere else. So this is a study done by someone else, Abby Swan at the University of Washington. She planted trees in the planted in computer simulations in the mid latitudes. And she looked at changes in precipitation. In this case, there is an increase in precipitation. So, but you could have the opposite effect. And this was done in the case of deforestation. Deforestation locally can increase runoff. In fact, in California, this is used as a way to do forest thinning to reduce the exposure of forest fires because it's has been an emergency recently. But there is also an increase in runoff, and the question is, whose water is that? But there is also, if you deforest a large area, you can change the weather patterns. You can affect, for example, the position, like in the other example, of the ITCZ, Intertropical Convergence Zone, and displace area of precipitation. We can often use this framework of rainfall recycling in the sense that the rainfall over this central area is contributed by water vapor that comes from local evapotranspiration, from evapotranspiration from adjacent areas and from the ocean. Now, if we deforest the surrounding areas, we might have an impact on this target area. So again, this is an issue of water tenure, which is even harder to define because, of course, we don't have a topography defining where water is going, only computer simulations. So lastly, I think we can, I will talk about pollution. Let's talk briefly, and then we end it here, about the irrigation paradox. So suppose that there is always the tendency of thinking that drip irrigation will solve the problems of the world. So now, when irrigation works in a very simple way, we withdraw water from groundwater from a river, we apply water on a crop, then part of this water is evapotranspired or consumed by crops, and part of it goes down as return flows, so to recharge the groundwater or rivers. Now, if a farmer different irrigation systems have different efficiency ratios between consumption and withdrawals. If we use surface irrigation, 50%, let's say, of the water goes down, is used by plants, and 50% is used goes back to the environment. So if a farmer used to withdraw 100, at least 50 were going back to the environment to recharge groundwater, for example. If we replace with drip irrigation and the farmer keeps withdrawing 100 units from the river, 95 go to the atmosphere. So the farmer will be happy, can irrigate more land or do a double cropping or something else, but the environment will suffer. So this is the so-called irrigation paradox. Because of the irrigation paradox, we have that the increase in efficiency leads to an increase in, doesn't lead to a reduction in consumption. So there is another problem here of water tenure, because if the farmer makes this replacement, the environment will suffer, or the downstream population or adjacent farms will suffer from that. And again, whose water is that? Because the farmer was allocated, typically the allocations are based on withdrawals, not on consumption. So this shows, again, how important in any form of water management to define a water tenure, to understand also that there are different mechanisms of appropriation of water. The typical example is by diverting water from a river. There are more subtle and less intuitive mechanisms, planting forests, changing crops, changing irrigation system, through which someone can take water from the environment or from somewhere else. And I think I'm running out of time. I had more things to show, but I think this is a good point for us to stop. And we can stop it here with where we started, that there are different uses of water. And sometimes it's a short blanket. And the other interesting point of discussion in these days should be in the moment in which we think we acknowledge both the human rights to water through a human right to food and the rights of nature. It's easy to make a public statement about them, but there are problems because since it's a short blanket, they collide with each other. They are in competition with each other, and so there is a big issue of adjudication of these rights and which one or what type of compromise can be found. Thank you.
Thank you very much.
Questions?
Thank you, Paolo, for the. Can you hear me?
Yeah.
Okay. Thank you for the nice presentation. I have a general question that maybe also for Cristina. It's related to the hydraulic infrastructures that are sustaining agriculture with irrigation. You show clearly that that we need to change the way in which water is used and distributed for agriculture, but we are still doing in all Western countries. We are still doing this by using old infrastructure that are being built more than 70 years ago, in a period in which people even know that there was groundwater, for instance. And now, and also in your presentation, in Christina's presentation, groundwater is taken like something which is okay. It is there, but maybe is not as a strong and important resource. This reflects 100 years of thinking of our society in a direction which the most important resource is surface water. This is not completely true, and I think that we need to open a new age, I would say, or a new period in which we think about water resources, combining these two possibilities. Let's say we can use also groundwater for storage, and not only reservoirs. And but this should be done in a proper way, and I think that we still not have the capability of doing that, and I'm closing, and also in part because the number of people that study in this field are declining. We are almost no more engineers, for instance, that are able to think in this way. So we need new engineers that are able to think in a new way about water resources, and we don't. I mean, we need that. I don't see in our universities new developments in this. So I would like.
No, I agree with. I think here I was referring indistinctively to groundwater and surface water for irrigation, because of course it's a big source of water for irrigation in many parts of the world. I think in the water sustainability debate there is always a lot of awareness about the declining levels of groundwater and the groundwater depletion problem. No, I was about to get there. Sorry. I didn't show that here, but most of the times I show that because when you look also climate change simulation, etc., there is the need for storage. We know that dams are controversial. We know that also the whole thing of little storage has already been implemented. And in California they did, and this is the slide I usually show, they show the total volume that you can have of storage in the existing surface reservoirs, and orders of magnitude more, I think it's 20 times more water can be stored in the groundwater. Some aquifers are ideal for that because they are not very close to the ocean. They are not discharging to the ocean directly, so they're really a good place to do that. And so in California, they are trying to advocate for that. Particularly in a climate where you have winter rainfall that is the rainy season is not compatible with the growing season. That's the ideal place. You don't have evaporative losses. You can, in fact, yeah. So they are called all managed aquifer recharges. I think the interesting thing there, and that unfortunately, because the speculation is this is the power of capitalism in a way, they created this new notion not only of water markets but of water banks. You can buy the water when the water rights are cheaper. You store it in below ground and you sell it when they are higher. So they are thinking about it. There is a system. You can check it out. There is literature on this. It's amazing.
Just a little comment. For the CO2, there are the credit of carbon. What about the credit of water? Because I mean, we are buying credit of carbon to do agriculture, especially in the United States.
Yeah.
What about buying credit of water for using water, for doing agriculture?
I'm not very convinced about this. I'm not convinced about the market-based solutions for the. I think this is something that is more of this neoliberal approaches to create, to convert an environmental problem into a business. And because but this is becomes at some point becomes more ideological than anything. But I don't think this addresses the problem, because every time you create a water market, you take water away from agriculture, you give it to another sector. Where there are more water markets in the US, if there is a water crisis, they pay the farmers to leave the land fallow and the mining prevails. So we can do that to some extent, but I don't think this can be very disruptive for a system that has also very small margins. And I'm thinking about the case of smallholders. But let's look at this other thing. There are two, in this discussion, we always need to be careful. Let's divide the water cost into things. There is a cost of provision cost, which means like the water supply in the system, the water is treated, there is the delivery, and there is pumping infrastructure. Okay, that's one thing. In agriculture, first of all, water is seldom treated, right? Only Saudi Arabia does, I was meeting with them the other day, they do some desalination, but it's rare, right? But let's assume that it is not treated. Then there are some delivery costs, and you would pay the irrigation consortium for that, maybe. But here, when people talk about, and someone needs to pay for those, so we need to find a model to. But here we are talking about making people pay for water by itself, which is different than is different from paying for food. Food is a manufactured good. Here we are talking untreated water, so we don't pay for air. So I don't know. At some point. But this is no. I know, but this becomes a governance problem. But sometimes also the sort of tragedy of the commons have been overemphasized to create to commodify pieces of nature. So perhaps there could be alternatives. Yeah.
So I have a point. We have to go
Yeah,
out.
sorry.
And I have another point. For sure, you have to come back. The discussion is open. You have to come back soon. So, yes.
Okay, I would like to ask PhD students to come with me to the ground floor for the photo shooting. And so we start with the lunch break. we are quite on delay. Would like to say to have lunch break. It is 1:30, 1 hour. So at 2:30, we come back here. Thank you. So PhD students on the ground floor. Yes, exactly.